Good afternoon, students, and welcome back to the chapter that is molecular basis of inheritance. And this is my last video regarding the question and answers of the main NCERT exercise. Clear? So the very first question is, group the following as the nitrogenous bases and nucleosides: adenine, cytidine, thymine, guanosine, uracil, and cytosine. Clear? So the nitrogenous bases are adenine, thymine, uracil, cytosine, and nucleosides are cytidine and guanosine. Clear? Then the second question is, if a double-stranded DNA has 20% of the cytosine, then calculate the percent of adenine in the DNA molecule. As you all know that the number of cytosine is equal to the number of guanine and the number of adenine is equal to number of thymine. So if cytosine is 20%, then guanine is also going to be 20%. Both of them, they are going to contribute for 40% of the total DNA content. So the rest left is 60%. Clear? So if we are saying that A is X and T is also X, then 2X is equals to 60, or X is equals to 60 upon 2. That is going to give us 30. Therefore, adenine here is 30% of the total DNA content. Getting my words? Then the next question is, if the sequence of the one strand of the DNA is written as 5-ATG, then again ATG, CA, TGCA, 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 TGCA up till 3-N, then what is the complementary? How is the complementary strand of the DNA going to run up in the 5- to the 3- direction? So accordingly, as we all know, according to the complementarity present in the DNA molecule, that if the one strand is running in ATG, CATG, CATG up till 3-N, then the complementary DNA strand will be G, C, A, T, G, C, A, T, G, C, A, T, G, C, A up till 3-N. Clear? Now, question number 4. If the sequence of the coding strand in the transcription unit is written as follows, that 5- is 5-ATG, CA, ATG, CA, ATG, CA, ATG, CA up till 3-N, then write down the sequence of the mRNA molecule. So answer over here is that 5- is equals to AUGC, 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 AUGC up till 3-N. Okay. Then fifth question is, which property of the DNA double helix led Watson and Crick to hypothesize semi-conservative mode of DNA replication and explain? As already I have explained you the semi-conservative nature of the DNA molecule in my topic, that was replication. Still I would like to give you all scenario of that. As we all know that according to the Watson and Crick model, DNA molecule, it is present in a double-stranded form which is helically wounded around the central core axis. Clear? So what happens over here, that during replication, these helices or these helical coils, they get unwounded and the DNA molecule, it becomes straightened up. Clear? And then what happens, the hydrogen bonds that are present between the nitrogenous bases, they break up, producing what? A replication fork. This sort of replication fork is produced in them. So what happens over here, that new strands, they are getting synthesized on the old DNA template strands, and after the complete formation of the DNA strands on the DNA template strands, what happens, both of them get separated and a new out of one DNA molecule, two new DNA molecules have been produced. And here the two new DNA molecules that have been produced, they are having one old strand and one new strand, one old strand and one new strand. Thus we say that DNA molecule is semi-conservative in nature. I have already discussed in detail over there in my videos. You can have a glance over it, or you can have a glance in your NCERT book. Then depending upon the chemical nature of the template, DNA or RNA, And the nature of the nucleic acid synthesized from it. List the type of the nucleic acid polymerases. So one is DNA dependent DNA polymerase, used as a DNA template for synthesis of the DNA. Then DNA dependent RNA polymerase uses a DNA template for synthesis of RNA. And 3, RNA dependent DNA polymerase uses RNA template for synthesis of the DNA, okay, in reverse transcription process. Now next question, that is question number 7. How did Hershey and Chase differentiate between DNA and protein in their experiment while proving that DNA is a genetic material? Clear? So I have already given my complete video on the Hershey and Chase experiment, that they have selected radioactive sulfur and radioactive phosphorus in their experiment, and how they have conducted their experiment. So I have already discussed in detail the whole of the experiment over there. Clear? Then differentiate between the following: repetitive DNA and satellite DNA, mRNA and tRNA, template strand and the coding strand. So when we are talking about the repetitive DNA and the satellite DNA, talking about the repetitive DNA, a DNA molecule consists of a large number of the base pairs arranged in a definite sequence. Some of these sequences are coding, called exons. Others are non-coding, called introns. So there are some specific regions in the DNA molecules where a small stretch of the non-coding DNA sequence is repeated several hundred number of times. The stretch of the DNA which is repeated several times is called as repetitive DNA. These repetitive DNA stretches are found at many places throughout the length of the DNA and are specific in each individual. These are said to be the repetitive DNA. And when we are talking about the satellite DNA, the repetitive DNA, when separated from the bulk genomic DNA by the technique of density gradient centrifugation, form minor peaks where the bulk DNA forms the major peaks. The minor peaks are referred to as the satellite DNA. Already this also I have given you in my videos. So kindly have a look at the videos. Okay. Now differences between the mRNA and the tRNA. mRNA is definitely known as the messenger RNA and tRNA is the transfer RNA. mRNA is synthesized in the nucleus on the DNA template strand, and tRNA it is present in the cytoplasm of the cell. Clear? mRNA, though it is synthesized in the nucleus, then later in the later stages it is transferred to the cytoplasm. Clear? Then the function of mRNA is to carry out the genetic coding or copy of the genetic codes present in the DNA molecule and transfer it to the tRNA molecule for protein synthesis. And what happens about the tRNA? tRNA reads up the codes present in the mRNA molecule, and accordingly it picks up the amino acids from the amino acid pool present in the cytoplasm and transfers the amino acids to the ribosomes for synthesis of proteins. Clear? Then template strand and all already I have given you all. List 2 essential roles for ribosome during translation. The 2 essential roles of ribosomes. Ribosomes provide the site for protein synthesis. I already have told you all about this. The mRNA comes in contact with the small subunit of the ribosome, and then protein synthesizing complex is formed. The large subunit provides sites for amino acid binding. Second is, ribosome also act as a catalyst for the formation of the peptide bond. Clear? Now, 10th question is, in the medium where E. coli was growing, lactose was added, which induced the lac operon. But why does lac operon shut down after some time after addition of the lactose in the medium? So the answer to this question is, lactose acts as an inducer by switching on or off of the operon. If lactose is present in the bacterial culture, it inactivates the repressor and induces the transcription for synthesis of enzyme, that is beta-galactosidase. Already that has been discussed in your videos. This enzyme acts on lactose so that the lactose is synthesized. Absence of lactose causes synthesis of the repressor mRNA that binds to the operator region and stops transcription. In this way, the lac operon is shut down. Getting my words? Now, 11th question. Explain in 1 or 2 lines the function of the following, that is the promoter, tRNA and exon. tRNA का तो अभी मैंने आपको explain कर ही दिया है. tRNA का function क्या है? Promoter की जब मैं बात करती हूँ, promoters का जो function है, promoter gene lies adjacent to the operator gene and marks the site where the RNA polymerase enzymes binds up. Clear? And talking about the exons. In split gene arrangement of the eukaryotic mRNA, the sequence of the bases that appears in preceded RNA are called as exons. They are interrupted by introns in unprocessed RNA molecule. Clear? Then 12th question is, why is the Human Genome Project called as a mega project? I have already discussed in detail what is Human Genome Project and why is it known as a mega project. That is, I have given you in detail over there. Then what is the DNA fingerprinting? Mention its application. That is also given in my videos, that what is the DNA fingerprinting and what are the various applications of DNA fingerprinting. Then briefly describe the following, that is transcription. Transcription is the process where mRNA is synthesized on the DNA. Then polymorphism, that is also I have explained. Then what is translation? Translation is the synthesis of the protein according to the mRNA molecule. And bioinformatics and all I have already discussed in detail with you. Clear? So here we have completed our chapter, and I have also discussed the question answers all along with you all. So with this we complete the chapter completely. Now you have to do the question answers, as I hope that you people are completing the question answers in your registers. If not, then kindly do it. Make a register, separate register, as you people have made it in the 11th standard, and start doing the question answers of every chapter. Clear? And I'll be giving you certain worksheets regarding the respected chapters. You have to complete the worksheets also, and you can send me the screenshots of those worksheets, and we are just going to discuss in detail the questionnaires or whatever I am sending in the worksheet in my online streaming classes. Getting my words? Till then, thank you, and we are just going to start up with the very next chapter, that is evolution, in my upcoming next videos. Till then, thank you.